Welcome to Med in 5. The goal of our channel is to simplify medical concepts and help you understand them like a boss. This video is part of our series explaining bacterial infections. Today, we're talking about non-lactose fermenting gram-negative bacilli. Before we dive in, think about these questions. Which organism causes typhoid fever? Which species causes an infection that mimics appendicitis in kids? Which swarms across agar plates and smells like burnt chocolate? We'll get back to them shortly. Non-lactose fermenting gram-negative bacilli are gram-negative rods that do not ferment lactose, so their colonies stay pale on McConkey agar. Many of them are intestinal pathogens, and several can produce hydrogen sulfide, giving useful lab clues. First up is Salmonella. There are many species, but the big ones are Salmonella typhi, which causes typhoid fever, and the non-typhoidal species like Salmonella enterocolitica, which are usually linked to contaminated food. Salmonella is motile, oxidase negative, and, importantly, it produces hydrogen sulfide. On triple sugar iron agar, the microbe's hydrogen sulfide production causes black discoloration at the bottom of the tube. Typhoid fever is due to Salmonella typhi or paratyphi, typically transmitted through contaminated water or food in regions with poor sanitation. As typhi penetrates intestinal M cells and spreads systemically, typhoid fever often starts with a stepwise fever, fatigue, and abdominal pain, and if untreated, it can lead to intestinal hemorrhage or perforation. Around the second week, patients may develop rose spots on the abdomen along with hepatosplenomegaly and, in some cases, a chronic carrier state in the gallbladder. Non-typhoidal species such as S. enterica, on the other hand, are often acquired from undercooked poultry, raw eggs, or unpasteurized dairy. They usually cause self-limited gastroenteritis. Fever, abdominal cramps, and diarrhea usually begin 6 to 48 hours after ingestion and typically resolve in a few days. However, severe dehydration or bacteremia may occur in vulnerable patients. No antibiotics are needed for uncomplicated gastroenteritis, but in severe or systemic cases, especially typhoid fever, empiric treatment includes ceftriaxone or azithromycin, with adjustments based on culture results. Next is Shigella. Shigella is non-motile, oxidase negative, and unlike Salmonella, it does not produce hydrogen sulfide, so the TSI slant stays yellow with no blackening. Shigella spreads easily in daycare and crowded settings through the fecal-oral route, often via contaminated food like fresh produce, undercooked seafood, and improperly handled salads. Infection begins with abdominal cramps, fever, and sudden diarrhea that becomes bloody with mucus, called dysentery. Dehydration and febrile seizures may occur, especially in young children. The bacteria invade M cells in the colon, spreading cell to cell and causing intense inflammation. Shigella uses IPA proteins to invade host cells and promote its spread through the colonic epithelium. Some strains produce Shiga toxin, which can cause hemolytic uremic syndrome or HUS, leading to anemia, low platelets, kidney failure, and serious complications. Treatment may involve azithromycin or ciprofloxacin in moderate to severe cases to shorten illness and prevent spread, though many cases resolve without antibiotics. Now, let's cover Yersinia enterocolitica. This one's often forgotten. It's non-lactose fermenting H2S negative and oxidase negative. On the surface, it may seem like the others, but Yersinia is urease positive and cold tolerant, able to grow in refrigerated foods like pork. Yersinia can cause fever, diarrhea, and right lower quadrant pain, mimicking appendicitis. This is due to mesenteric lymphadenitis, especially in children. Severe infections or immunocompromised patients may require doxycycline or fluoroquinolones, though mild cases often improve without antibiotics. Next is Proteus mirabilis. This species is famous for swarming motility. It moves in waves across agar plates, which makes its colonies look like ripples. It's urease positive, raising urine pH and increasing the risk of struvite stones. Proteus is oxidase negative, H2S positive, and causes UTIs, particularly in catheterized patients or those with structural abnormalities. It has a distinctive fishy or burnt chocolate odor, which is sometimes used as a diagnostic clue. Treatment depends on susceptibility, but options often include beta-lactams or fluoroquinolones, especially for resistant strains. Let's go back to our questions. Which organism causes typhoid fever? Answer: Salmonella typhi or paratyphi. Which species causes an infection that mimics appendicitis in kids? Answer: Yersinia enterocolitica. Which swarms across agar plates and smells like burnt chocolate? Answer: Proteus mirabilis. Got them all right? Yay! If this video helped you understand non-lactose fermenters, hit like, share, and subscribe. Tap the link below to download the summary table comparing their microbiology features and clinical relevance. Thanks for watching.